Today we're going to be talking about one of the most fascinating and potentially life-saving areas of modern medicine, transplanting genetically modified pig organs into humans. This process is called xenotransplantation, and just a few years ago this sounded like science fiction, but today it's slowly becoming a reality. A recent New York Times article written by Karen Robin, along with reports from many medical centers, described a pig kidney functioning in a human body for an extended period of time without immediate rejection. This marks a major milestone in transplant history. Why is this so important? Because every year, thousands of people die waiting for organs that never arrive. Today, I'm going to be talking about several key areas. First, why pig organs are used, the science behind xenotransplantation, what we found from studies, and the future of this field. We will also talk about the challenges, ethical considerations, and the exciting potential for a medical revolution that would change organ transplantation as we know it. Let's begin by examining the global organ shortage. Each year, the demand for organs far outpaces the supply. That means many die of kidney failure and some wait between 5 to 10 years on dialysis without receiving a transplant. Kidneys are the most needed organs. Diabetes, hypertension, and infection all contribute to this rising need. Why is the shortage so severe? Donor organs are unpredictable. Not every organ is viable, and even when one is available, matching blood type, tissue type, and immune compatibility is difficult. The key takeaway is that doctors have a skill to perform transplants, but the limiting factor is supply, not expertise, and this is why xenotransplantation is gaining serious attention. Imagine a world where organs could be produced on demand. People won't have to wait years for organs and countless lives would be saved. This is what makes xenotransplantation a potentially revolutionary solution. Such technology could reduce the strain on healthcare systems, lower costs associated with long-term dialysis, and improve quality of life for the patient. Additionally, think about the psychological relief for the patient and the family that comes from knowing that an organ can be available when it's needed. Xenotransplantation is a transplantation of organs and tissues from one species to another, in this case from pigs to humans. But why pigs? The organ size closely matches that of humans, they can breed quickly, produce large litters, and they can be raised in controlled, sterile environments. They also have a previous history of medical use, for example in insulin. The main challenge is the human immune system. It identifies pig organs as foreign and attacks them immediately. This is known as hyperacute rejection, and it can destroy an organ within minutes. However, with advances in gene editing, scientists can now modify pigs so that their organs are less likely to be rejected by the human immune system. This is opening doors to many more successful treatments, and it could potentially expand beyond kidneys, offering solutions to many life-threatening conditions. Early attempts in the 20th century mostly failed. In the 1990s, the first pig-to-human heart transplant failed within a few hours. In the 2000s and 2010s, improvements in immunosuppressants helped, but they were not sufficient enough to prevent organ rejection. The breakthrough came with CRISPR gene editing. Scientists could now remove genes from pigs that were responsible for immune reactions and replace them with genes from humans, protective genes. Now, they tested this in some brain-dead patients, and it showed that they could work in human bodies for short periods of time without immediate rejection, and this offers proof of concept. The New York Times article we mentioned earlier highlights one of the longest and most successful cases ever recorded, a true milestone in medical history. Now, we'll also talk about how these early trials will pave the way for future trials on living humans and what lessons we can learn regarding organ compatibility and immune management. Here's what the article showed. A genetically modified pig kidney was transplanted into a living human patient, and it produced urine immediately and continued functioning from weeks to months. This represented a first in xenotransplantation history. Experts were astonished. There was no hyperacute rejection, no clotting problems, and no immediate failure. This was a potential turning point in xenotransplantation. This success is the result of a combination of advanced genetic engineering and new immunosuppressive drugs. Now, it's very important to remember that this is still early research. We still don't know the long-term effects of this. However, reaching this level of early success is a crucial milestone. Creating genetically edited pig organs is a detailed multi-step process. The first step involves removing some pig antigens, like GGTA1 and CMAH. Removing these reduces the foreign signal that the organs send, increasing compatibility. Second step involves adding some human genes like CD46 and CD55. You can also add HLE genes to increase compatibility. Now, doing this, along with adding some other organs, increases compatibility and decreases the likelihood of rejection. Step 3 involves addressing risks. Pigs naturally carry retroviruses, which can be disabled with CRISPR technology. Step 4 involves raising and breeding these animals in secure, virus-free environments with rigorous screening to ensure organ viability. These steps collectively create organs that are more like human organs, which dramatically increases the chance of successful transplants. We'll also talk about how these developments can impact future research, including personalized organ modifications tailored to patients' needs. The transplant procedure itself is similar to that of a standard kidney transplant. The blood vessels are connected and urinary flow is established. After the surgery, the kidney turned pink, which indicated proper blood flow, and urine was produced within minutes. Lab tests also confirmed proper filtering function. Medications used included the anti-CD40 antibody and steroids. Now, surprisingly, the immune reaction was milder than expected, and this demonstrates that the gene edits were effective and the immune response was manageable, as well as the organ acting like a donor organ. The benefits of xenotransplantation are enormous. It could basically end the organ shortage with a virtually unlimited supply of organs. It can enable standardized organ production, and it can also extend to other organs such as hearts, livers, and lungs. If successful, it could revolutionize transplant medicine globally, drastically reduce waiting lists, save countless of lives, and reduce the burden on healthcare systems. We'll also talk about the economic impacts of having widespread organ availability and how this would change the face of healthcare delivery systems. There's still medical risks, including long-term rejection, chronic inflammation, hidden infections, and cloning issues. Ethical concerns include using animals for organs, the welfare of these animals, and the concept of designer organs. Regulatory challenges include determining the number of required clinical trials, long-term patient monitoring, and preventing disease from spreading from the pig to the human. Now, despite these challenges, scientists believe that the potential benefits could change medicine forever. Also, current debates in medical and ethical communities need to be considered as public perception matters. We're witnessing one of the most significant breakthroughs in medical history. Xenotransplantation, once considered impossible, is now slowly becoming a reality thanks to gene editing, controlled breeding, and advanced medication. A pig kidney functioning in a human signals a future without an organ shortage. If progress continues, pig organs will become a routine part of medicine within this decade. Thank you for watching this video. I hope you found it useful. If you have any further information or experience regarding this topic, please let us know in the comments.